Hello students, in today's video we see a cholinergic system and drugs. In previous video we have seen the classification, organization and function of nervous system. There we have seen that autonomic nervous system is being divided into parasympathetic and sympathetic nervous system. So basically, according to the action, the parasympathetic or sympathetic, whether it is acting as an agonist or antagonist, they are classified into four. If the drug produces the sympathetic-like actions, which is acting as an agonist, is known as parasympathomimetic, is also known as cholinergic system, or the drug that opposes the parasympathetic action as the antagonist, it is known as parasympatholytic or known as anticholinergic drugs, or a system that drugs which produces a sympathetic like action or agonist is known as sympathomimetic, is known as adrenergic system because the adrenergic is the primary neurotransmitter which is releasing from the sympathetic nervous system. Drugs that opposes the sympathetic like antagonist, it is known as sympatholytics, also known as anti-adrenergic system. Basically, in this video, we are focusing on cholinergic transmission, cholinergic neurotransmitter. Cholinergic neurotransmitter is the acetylcholine, which is a major neurohumoral transmitter, autonomic, somatic, as well as central sites. It has virtually no systemic therapeutic applications because its actions are diffuse and its hydrolysis catalyzed by both anticholinesterase and plasma butyrcholinesterase is rapid. In the cholinergic system, acetylcholine is a neurotransmitter which is a choline ester. It is a neurotransmitter of parasympathetic system, a nerve which synthesizes, stores, and releases the acetylcholine, which is also known as cholinergic. There are different sites of acetylcholine release where it releases. First is a ganglia or postganglia. Ganglia or preganglionic fibers of the autonomic nervous system, both in sympathetic and parasympathetic ganglia. This is a diagrammatic form where the acetylcholine is releasing and where a non-adrenaline or epinephrine, different other adrenal releasing neurotransmitters. In a skeletal muscles, we see only so on a somatic system acetylcholine release. Okay. In cholinergic pharmacology, we deal with the drugs acting on the synthesis, release, and action of the acetylcholine on both muscarinic and nicotinic receptor. Here we will see first how the neurotransmitter acetylcholine is synthesized, released into the synaptic cleft, and how it acts on the muscarinic and nicotinic receptor, and also the drugs that inhibit the acetylcholine synthesis and release. In next video, we will specifically talk about the drugs that act agonist and antagonist on muscarinic and nicotinic receptor. Starting with the synthesis of acetylcholine, first choline is reuptaken into the nerve terminal where it attaches the acetyl coenzyme A in the presence of enzyme cholinesterase, choline acetyl transferase, and thus form acetylcholine. This acetylcholine is then stored in a storage vesicles. This is a storage vesicles which upon receiving a nerve and action potential leads to a calcium efflux, and thus the vesicle releases the acetylcholine into a synaptic cleft. Okay, the acetylcholine release will then act on muscarinic or a nicotinic receptor for the further action. As we know, neurotransmitter should be brief or the receptor get desensitized. That's why we an enzyme called acetylcholine esterase which degrade the acetylcholine into the choline and acetate. This is the acetylcholine esterase which degrade the acetylcholine in choline and esterase, and this choline is reuptaken into the nerve terminal and is reused in the synthesis of acetylcholine. After seeing this process of synthesis and the release of acetylcholine, we see two drugs which act on the synthesis as well as on the release of the drug. First one is the hemicolinium. Hemicolinium, as the name suggests, hemi, which means half, so it is half as good as choline, and so it hits it for being better and says that it I cannot go inside and choline cannot go inside as well. That's why it inhibits or blocks the choline uptake into the exoplast, and thus inhibit the acetylcholine synthesis. Hemicolinium has no use clinically as it is so full of heat. So the next is the vesamicol. Vesamicol is a transpose of acetylcholine into the synaptic vesicle is blocked. Third is the botulinum toxin, which inhibit the acetylcholine release and relieve the spasm in spastic disorder, facial wrinkles. So, as like hemicolinium, botulinum toxin has some clinical benefits. After the synthesis, release, and the storage of the acetylcholine, we see the metabolism of acetylcholine. Basically, you have seen the acetylcholine is hydrolyzed by the enzyme known as acetylcholine esterase to the choline and acetic acid, and thus its action are terminated, and the choline is then recycled in acetylcholine biosynthesis. So therefore the T half of the acetylcholine is very short. Basically, though the choline esterase, which is is divided into true acetylcholine esterase, or we can say a plasma choline esterase. True acetylcholine esterase is basically bound on the membrane enzyme which is present in the cholinergic synaptic cleft and hydrolyzes the acetylcholine and other acetyl ester. The example of true acetylcholine esterase is the methacholine, and second cholinesterase is a plasma cholinesterase which is synthesized in liver and found in the plasma and in intestines, and it hydrolyzes the benzoyl and butyrylcholine esters, and the genetic variations are commonly seen with these enzymes. Thus, we have seen after synthesis of acetylcholine, it releases into the from the synaptic cleft and attaches to the neurotransmitter, neuroreceptor. Cholinereceptors are of two types, muscarinic and nicotinic. The muscarinic receptors are G protein coupled receptors, and it can be divided into five groups: M1, M2, M3, M4, and M5, whether where M3, 1, 3, and 5 are located on the GQ, and M2 and M4 are diseased and a cyclic AMP and located on a GI. And clinically, we use M1, M2, M3, and M4. M4 and M5 are not clinically in use. Secondly, is a nicotinic receptor. It is a ligand-gated cation channel divided into NM and NN receptor. Here we can see the subtypes and the characteristic of cholinereceptors, where it are secreted and what are the function of each cholinereceptors. M1 is secreted from the secretory glands like salivation, stomach acid, sweating, lacrimization. Second subtype is M2 of M receptor. From it is secreted from heart, and the action is decrease the heart rate in the case of bradycardia. M3 is present on a smooth muscles, on a gastrointestinal or gastric upper respiratory problems, respiration, and the characteristic is contraction. It contracts the smooth muscles like during the diarrhea, bronchospasm, or in a urination. M3 is present on a pupil and ciliary muscles receptors. It contracts and causes a meiosis during an increased flow of aqueous humor. Second is a nicotinic receptor. First is NM, which is located on a skeletal muscle and plate, and it acts on contraction of skeletal muscle. And nicotinic N receptors is located on a autonomic ganglia or adrenal medulla, and control the secretion of epinephrine and control the autonomic nervous system. So that's all for this video.